So we are continuing on our study of what these genes actually are. And it's going to take a very long time to get through the entire story. But where I left off, we talked about mitosis, this process of cell division where the chromosomes appear, move around, and then divide up. And we just finished talking about a second process called meiosis. It will stand you in very good stead if you know how to recognize the stages of mitosis. You need to know interphase is when mitosis is not happening, and then comes prophase, metaphase, anaphase, telophase, PMAT, if you like. And then you don't have to memorize every last step of meiosis, but you need to know it is a double cell division called reduction division, in which you end up with cells in which the number of chromosomes is cut in half. Who cares? Mendel had worked out that genes come in pairs, right? You've got two copies of each gene, each of which can be in one or more alternate forms called alleles. And you inherit one of each pair of genes from each one of your parents, and then you pass one of each pair of genes to each one of your offspring. And this is not just true in pea plants, it's true for the majority of eukaryotes with a few occasional funny wrinkles, including yourself. And now here's a bunch of microscopy geeks in the 1870s and 1880s who've worked out this dance of the chromosomes. And it looks like meiosis is doing the same thing. The point of meiosis is to ensure that you get one of each pair of chromosomes from each parent. With one cool exception that I'll get to today. You have pairs. Each one of your chromosomes comes in pairs. So you've got 46 chromosomes, which can be arranged in 23 pairs. You got one of each pair from each of your parents. And if you have kids, you'll pass on one of each pair to each of your kids. And we know that if meiosis goes wrong, you can end up with an egg or sperm that either has a missing chromosome or an extra chromosome in a pair. And that leads to fertilized eggs or zygotes that either don't develop or they don't develop as they should. So this is important. The reason why I dragged you through all of that cell division stage stuff is that genes and chromosomes are following the same rules. Could there be some sort of link between them? Well, you might remember that Mendel's work was forgotten about for 35 years, but it was rediscovered around 1900. And by 1902, a couple of scientists, don't worry about these names, Henry Sutton and Theodore Boveri, had proposed independently of each other that genes must be carried on chromosomes. Genes must be physically not the same thing as chromosomes, but located on chromosomes. This drew the attention of a professor at Columbia University in New York City, a gent named Thomas Hunt Morgan, who got interested in this problem. And he and his students started studying genetics, but instead of peas, they used fruit flies, Drosophila melanogaster. And again, this comes back to something I talked about earlier. If you want to study a problem in biology, it helps a lot if you can pick a particular species of living thing that's easy to handle for the things you need to do to study the problem. So Mendelian genetics applies to both fruit flies and elephants, but elephants take up huge amounts of space. They're very expensive to feed, and it takes, pregnancy lasts a year and a half. So it would take a very long time to do breeding experiments on elephants. Fruit flies are easy to raise. You can do it they used to use just glass milk bottles. They're cheap to raise. You can raise them on rotten or overripe fruit. You can raise millions of them in one room. It only takes you a week to go from one generation of fruit flies to the next. And they also have lots of traits that follow simple Mendelian rules, traits involving eye color, wing shape, body color, body shape, lots of different traits that could be shown to follow, to behave just like Mendel's laws. D. melanogaster, by the way, also has only 4 pairs of chromosomes. These are fruit fly chromosomes that you see, the stripy ribbon-looking things. In certain cells, the cells of the salivary glands of all things, the chromosomes happen to be unusually large. They're strongly banded. You can see that each one of those ribbons has what looks like stripes on it, almost like a barcode. And so they're relatively easy to study. They're easy to see, easy to map, easy to trace the inheritance of complicated banding patterns. And so fruit flies are a great example of a model organism, which is just a species in which something that you would want to study turns out to be particularly convenient to study. And to this day, fruit flies are used in fundamental genetics research all over the world. Here's Sarah Palin complaining that US government money goes to projects like fruit fly research in Paris. And that might not seem like a particularly important thing until you remember that fruit flies are a model system that's been used for over 100 years. We know a humongous amount about fruit fly genetics, and what we learn about fruit flies can be applied to all eukaryotes, ultimately including ourselves. Fruit flies have a lot more to do with you than you might realize. And in 1909, one of Morgan's grad students, a guy named Calvin Bridges, while sorting through a bunch of fruit flies that he'd bred, discovered one that had a trait he'd never seen before, a fruit fly that had white eyes, as seen on the right, instead of the normal eye color in fruit flies, which happens to be bright red. I'll talk later about exactly what causes this to happen. For right now, let's just say that Cal Bridges had discovered a mutant fly. And when we have a mutation, it does not necessarily confer the ability to shoot optic blasts or anything like that. It's not quite as spectacular as what the X-Men have. It's just a case in which an allele can suddenly change to a different allele in a single generation. And we will later talk about why that's the case. You've seen one example already, if you remember that cat with curly ears that we talked about back when I was talking about artificial selection. That was a cat with a mutation, a change in one of its genes, creating a new allele that the cat could pass on to future generations of kittens. This is another example of a mutation. So Bridges took his mutant fly and started breeding it. And he found that if you crossed the white-eyed fly with a purebred red-eyed fly, purebred meaning it's descended from red-eyed flies for as long as anybody cares to check. If you cross a purebred red-eyed fly with a white-eyed fly, the offspring all have red eyes. So red eye color is dominant to white eye color. By the way, in fruit flies, we don't do this for all species, but in fruit flies, the traits that are by far the most typical in natural populations are called wild type. If you were to leave a bunch of bananas out in your kitchen for a little bit too long when the weather is nice and warm, you might well get fruit flies in your own kitchen, and those are all going to have red eyes. I've actually trapped fruit flies buzzing around the dumpsters back of the Christian cafeteria, and those always have red eyes. Red eye is by far the dominant phenotype in nature, so we call that wild type. Variable species may not have one single wild type, but fruit flies, we can define wild type. And so if you cross two of those red-eyed flies from the F1 generation, you get 3 red-eyed flies for every 1 white-eyed fly, and that's exactly what Mendel's laws would predict. So far, this is following exactly the same rules as, purple and white flower color in pea plants. Except Bridges noticed something weird. All of the white-eyed females in that F2 generation were males. And by the way, male and female fruit flies, it takes a little practice, but it's possible to tell the difference between them. Slightly different proportions in the abdomen, different color schemes, and males have some special bristles on one of their legs called the sex combs. So with a little practice, it's pretty straightforward to tell a male and a female fruit fly apart. So when he did that cross, where you got 3 red-eyed flies for every 1 white-eyed fly in the F2 generation, half the males in the F2 generation were red-eyed and half the males were white-eyed. None of the females were ever white-eyed. So this is weird because we've got a sex distinction here. It doesn't really apply to pea plants where each flower produces both eggs and sperm in the same flower. But it's something odd going on here in fruit flies. Why should there be that kind of sex bias when it comes to inheritance of eye color? You could get a white-eyed female if you crossed a white-eyed male with one of the F1 females. And if you did that, a quarter of the offspring would be white-eyed females. But then if you crossed a white-eyed female with a red-eyed male, you get half the offspring are red-eyed females and half the offspring are white-eyed males. What is going on here? Here's our exception to the rule that chromosomes always come in pairs. In many organisms, not all, but including humans, including all the mammals I can think of, and by good great luck also including fruit flies, one of the pairs of chromosomes varies depending on the sex of the organism that has them. And what we call sex chromosomes determine the individual's sex. By the way, just so we're clear, sex is not the same thing as gender. Sex refers to what chromosomes you've got and what plumbing you've got. And incidentally, we probably won't have time to talk about why, but there are some interesting cases where the chromosomes that you get actually don't match up with the plumbing that you've got. There are people with intersex conditions that can have in which they can have one set of sex chromosomes, but not the anatomy that you would expect. And that's more common than people realize. Gender has a lot to do with the society you're in, the role you play, and the nature that you feel yourself to have. And that is a much more complicated thing than biological sex is. So chromosomal sex is one thing. Gender roles and gender identity are something else. And we really do not fully understand how those relate to each other or why it is that there is sometimes a mismatch between the chromosomes you've got and what you feel yourself to be. That's just one of those things. You know, people are strange and complex and beautiful creatures, and that's just the way stuff goes. Anyhow, and as I said, you can have mismatches sometimes between what chromosomes you've got, what anatomy you've got, and what you feel yourself to be. This is a more complicated field than we can get into. But when I'm talking about sex here, I'm usually meaning what chromosomes you've got, with the caveat that you do get some exceptions. In most mammals, and also in fruit flies, females have 2 copies of a large chromosome called the X chromosome. Males, however, will typically have 1 X chromosome and 1 smaller chromosome called the Y chromosome. This is not universal, by the way. In birds, it's actually reversed. In birds, females are XY and males are XX. Although technically they use different letters, so we'd actually say that female birds are WZ and male birds are WW. Lindell, could you mute? Lindell, could you mute, please? In other critters, like in turtles and crocodiles,
sexuality is a body choice.
Sex.
Why do men think that? Oh my gosh, that's a coochie. Yeah.
It's just imagine. It is straight. She finna come back. You ain't even gotta worry about it.
I'm not getting that.
All right, Lindell, mute, please. Okay, I'm sorry about that. I can't reach out through the screen when I'm doing slide share like this. All right. Okay. So in most mammals and in fruit flies, females have 2 X chromosomes and males have an X and a Y. In birds, it's the reverse. In turtles and crocodiles, your sex is not determined by your chromosomes at all. It's determined by the temperature the egg is incubated in. Quite a number of fish actually switch sexes during the standard lifespan. Clownfish, for example, all start out life as males, and then when they finally reach maturity, they switch over to females. Which means that if Finding Nemo was real, Marlin would be trying to get Nemo back while negotiating the fact that he was transitioning to Marlena, which I think would make a much more interesting movie, but they didn't ask me. So this is not biologically universal, but it is the case in most mammals and in fruit flies. Here's some human female chromosomes. Again, you can see that these have banding patterns that match up, and the members of each pair are pretty close to the same in length. And you'll notice that there are 22 pairs of non-sex related chromosomes and 1 pair of X chromosomes right down there. And by the way, any chromosome that isn't a sex related chromosome is called an autosome. And there's chromosomes from a human male. The autosomes look the same in males and females. We've all got 22 pairs of autosomes, but the male has 1 X chromosome right there and 1 much smaller chromosome called a Y chromosome. I'll mention, and I wish I had time to talk about this more because at least one of you was asking questions about this that were really interesting. We all develop, every one of us, when we start developing a genitourinary system, it looks the same. We all have a pair of tubes and a pair of little lumps of tissue, and we've all got similar genitals between our legs that are not either exactly male or female, but look more female than anything else. If you have a Y chromosome, the Y chromosome has a region on it called SRY. And I don't have time to explain how this works, and I don't know that I fully understand how it works, but the SRY amounts to a kind of switch that you flip. And if an SRY section of a Y chromosome is present, the plumbing that you've got when you're an embryo will usually begin shifting towards the male pattern. The lump of tissues, the two lumps of tissue will drop down and end up as testes. The pair of tubes that would become a uterus stop developing. What would be your labia actually grow together and become a scrotum, and your clitoris and your urethra enlarge and end up becoming a penis. If you don't have an SRY present, then those two lumps of tissue stay inside and they become ovaries. The tubes that would have become sperm cords never develop into anything much. A different set of tubes develops into uterus and fallopian tubes, and your labia stay open and the clitoris does not become a penis. And the switch that determines which of those two patterns you follow is located on the Y chromosome. There are some interesting instances where it doesn't work. You can actually have people whose anatomy is female, but they've got an X and a Y chromosome. And you can have people that have two X's but develop as male. There's even one group of people in Guatemala with, it's called, I think it's 5-alpha reductase deficiency, where some of the people look female until they hit puberty and then suddenly transform into males. It's a condition called guevedose, which is Spanish for getting your balls at the age of 12. So fascinating stuff, really cool stuff. But this is the pattern that we see by far the most commonly in our own species. And Thomas Hunt Morgan had gotten the idea to study fruit flies from Nettie Stevens, a professor at Bryn Mawr College in Pennsylvania, which is where he worked before he got the gig at Columbia. She had discovered that many insects, including fruit flies, have this chromosomal sex determination, where the females have two X chromosomes and males have an X and a Y. And she might have gone on to make some of these discoveries before Morgan did, but she actually died tragically young, unfortunately. But she was able to contribute this to the puzzle. Okay, the question is, can you still be intersex if you don't have a vagina or a closed vagina? The answer is there are a lot of different intersex conditions. I've read of a case of one where a boy was outwardly male, except he started getting bloody urine every month because it turned out he had an internal uterus that emptied right into his urethra. So you can get things like that, but I'm not a doctor who specializes in this sort of thing. You probably be best off consulting a medical specialist for detailed questions on intersex conditions. But yeah, there's all sorts of possible outcomes that can happen. Okay, so what's this got to do with fruit fly eye color? Morgan hypothesized that the reason why you got that weird sex bias when you crossed red-eyed and white-eyed fruit flies must be that the genes are physically being carried on specific chromosomes. Specifically, the gene for eye color must be physically located on the X chromosome. The sex chromosomes do more than just determine your sex. You know, there's a lot of physical traits that have genes located on the X chromosome, but they don't have anything to do with whether you develop a hoo-ha or a tallywhacker. Eye color's got nothing to do with, you know, with your sex or anything like that. So the sex chromosomes are not entirely accurately named. But what must be going on here is that this eye color gene must be physically located, or as we say, carried on the X chromosome. And that's a phenomenon that we call sex linkage. And here's how it works. The original white-eyed fly was a male, and we'll write its genotype as X with a little superscript W, Y. We can write the female as XX. She's got 2 X chromosomes. The male has an X and a Y. And we often use a plus sign to mean wild type, to mean the phenotype that you would by far most commonly see in nature. And I'm using these pluses here because it can be hard to tell whether a superscript W is uppercase or lowercase. So our original white-eyed fly has 1 X chromosome that's carrying the white-eyed allele and 1 Y chromosome, which is not carrying any alleles for eye color. The X and the Y do not carry the same alleles. The female is red-eyed and descended from a long line of red-eyed flies, so she's presumably homozygous for the wild-type red color. And this is the Punnett square that you get. Half your offspring are heterozygous females that have red eyes because red is dominant to white. And half of your offspring are red-eyed males. Okay, you with me there so far? Notice that the male offspring of the white-eyed fruit fly are not inheriting his white-eyed allele. The male cannot pass on his X chromosome to his male offspring because if he did, they would be his female offspring. Now, if you take a male from the first cross right over here and a female from the first cross, what happens? Well, 1/4 of the offspring are homozygous dominant red-eyed females. They have 2 X chromosomes because they're female. And both of these are carrying a copy of the wild type allele. Those are just red-eyed females. 1/4 of these offspring are going to look red-eyed females, but they're carriers, they're heterozygous for it because they've got 1 copy of the wild type allele on one of their X chromosomes and 1 copy of the white-eyed allele on the other X chromosome. 1/4 of the offspring are going to be red-eyed males. They have 1 X chromosome that has the red eye color and the other chromosome is a Y. And 1/4 of the offspring are the white-eyed males because they've got 1 X chromosome that's carrying the white-eyed allele and they've got 1 Y. That explains the result that you get. 1/4 of these offspring are going to be white-eyed males. 1/4 will be red-eyed males. And 1/4 will be homozygous red-eyed females. And 1/4 will be heterozygous red-eyed females. That matters because that clinches the case that genes are physically carried on chromosomes. and that Mendel breeding his peas and microscopists of the 1870s and 80s staining cells with dyes had actually been working on two sides of the same jigsaw puzzle. The pieces are starting to fit together. Here's a kitty cat example that I have to mention because, you know, I have to mention kitty cats. The gene for orange color is sex-linked, and it's dominant to the gene for not having orange color. Orange is dominant to no orange. Incidentally, if an orange gene is present, that orange gene will block the appearance or will block the appearance of the gene for black or brown color. That's more epistasis. Remember, we can have one gene that modifies or even blocks the effects of another gene. So a male cat can either be XY carrying the orange allele, in which case they will look orange, or they will be X not orange, Y. Their X chromosome will either be carrying the orange allele, in which case they'll look orange, or their X chromosome won't be carrying the orange allele, in which case they won't look orange. They might look black, brown, gray, tan, what have you. Females, however, can be heterozygous. And if you have a heterozygous female cat, the cat develops patches of orange and not orange fur, like this cat seen here. This is called a tortoiseshell cat. For our purposes, you could think of this as a form of incomplete dominance. It's actually due to a really neat phenomenon called selective X inactivation that I just don't have time to get into. But for our purposes here, we can think of this as a case of incomplete dominance, where the orange allele does not completely cover up the presence of the not orange allele, and you end up with an orange and not orange spotty cat. And this pattern is called tortoiseshell. Incidentally, tortoiseshell cats are almost always female. There's almost no way to get a male tortoiseshell cat unless you happen to have a cat with two X's and a Y. And that happens because of an error in meiosis, and the cat is sterile. You get a condition called Klinefelter syndrome if you end up with two X's and a Y because of an error in the meiosis that produced the egg or sperm that you came from. But that's pretty rare. So virtually all tortoiseshell cats you'll ever see are going to be female. They have to be. And incidentally, if you have a cat that's heterozygous for orange and also is carrying genes for white spotting, which we talked about last week, you have a calico. Calico cats have mixtures of orange, non-orange, in this case black, and white. So this is what a calico cat looks like. And calicos, like tortoiseshells, can only be female. You can get more complex things than that. So one of my cats, well, our cats, one of the cats that owns us at my house is actually a calico cat who also has the Siamese pattern and only shows her orange and black spots on the cooler parts of her body. So it can get more complex than that. For right now, just focus on the fact that this is a cat example of a sex-linked gene, one that's carried on the X chromosome, and one that doesn't directly have anything to do with sex. It has everything to do with fur color. If you want a human example, the most common type of color blindness is red-green color blindness, and you can detect blue and yellow, but red and green both look kind of gray. If you've got normal color vision, this picture right here, you should be able to see the numeral 12 quite clearly. If you have red-green color blindness, this is just going to look like a bunch of grayish circles, and you're not going to be able to see the numeral 12. And if you have this, it can be kind of annoying, especially if you have to, dress yourself or deal with stoplights. But you can lead a pretty normal life with this. This is not debilitating at all. And odds are that somebody in this class has it, because about 7% of all men in the US have it, but only 0.4% of US women. And the reason is that for a woman to have it, she has to be homozygous recessive. She has to have 2 X chromosomes, both of which are carrying the color blindness allele. If you're a man, you only have to have 1 X chromosome that has the color blindness allele. And a woman can be heterozygous. She can have 1 normal X chromosome and 1 X chromosome with the color blindness gene, and her color vision will be fine, but she can pass on color blindness to roughly half of her male offspring. So that's a human example that may be familiar to some of you, for all I know. It's a very common trait in humans. Little bit of a history lesson here for you. Here's Queen Victoria of England, born in 1819 and died in 1901. She was quite healthy and she had a lot of kids, but she passed on a sex-linked allele on her X chromosomes that conferred the phenotype of having a type of hemophilia. Specifically a type called factor 9 hemophilia. There's more than one type, but this is a pretty common type. Hemophilia is a condition where the blood does not clot, or if it does clot, it's very, very slow. Hemophiliacs can lead a more or less normal life, but they have to be very careful because a cut that a normal person would just shrug off can be potentially life-threatening for a hemophiliac. If they start bleeding, they can't stop, or at least it takes them a very long time to stop the bleeding. Queen Victoria was heterozygous, we now know, for hemophilia. Sorry, just checking the chat. And here's her family tree. So she, being female, had 2 X chromosomes, and one of them was carrying a hemophilia allele, and I'll write that as X with a little h. Prince Albert, her beloved consort, Albert of Saxe-Coburg-Gotha, being male, had an X chromosome and he had a Y chromosome. Okay. So Edward VII must have gotten a Y chromosome from Albert and an X from Queen Victoria. By the way, a woman always contributes one of her X chromosomes to her offspring. The male contributes either an X or a Y. It's basically the father that determines the sex of a child, although there's no real way to do this consciously. But Edward VII right there, by luck, got Queen Victoria's normal chromosome. So he was XY, and he did not have hemophilia. And so he did not pass it on to the British royal family. So King Edward had George V, and George V's son was George VI, and George VI's daughter was Queen Elizabeth, who's queen now. And she had Prince Charles, and he had Prince Harry and Prince Meghan and whatever the heck it is. I don't really follow. But there's no history of hemophilia in the present British royal family. Her son Leopold, on the other hand, got a Y from Albert, and just by luck happened to get Queen Victoria's hemophilia chromosome. He was a hemophiliac, and he passed it on to his daughter Alice, and she passed it on to her kids, including Viscount Trematon over here, who was a male hemophiliac and died at the age of 20. Queen Victoria's daughter, Alice, right here. She got an X from Albert and an X from Victoria, and just by luck, she got Victoria's hemophilia gene. She's normal, but she passed it on to her daughter over here, Alexandra, who got a normal X chromosome from her father, I forget who that was, and a hemophilia chromosome from her mother, Alice, who got it from her mother, Queen Victoria. Alexandra married this guy, Tsar Nikolai II, the last emperor of Russia, and he's XY. And they had 4 daughters, Olga, Tatiana, the one I can't remember, and Anastasia. You might have seen that animated film, Anastasia. Well, that's her. She's one of those. And then their youngest kid and only surviving son, Tsarevich Alexei, right there, was a hemophiliac. He had, just by luck, gotten the hemophilia-carrying X chromosome from his mom and a Y chromosome from his dad. And he would have been the next emperor of Russia, except the communists took power and the entire royal family ended up getting shot. But that's why the crown prince of all Russia was a hemophiliac boy. He'd gotten it from, just by stupid, rotten bad luck, from his great-grandmother, Queen Victoria of England. Incidentally, Queen Victoria's granddaughter, Victoria Eugenie, was a carrier. 2 of her brothers and 2 of her sons were hemophiliacs. This is her son, Prince Gonzalo, who was unfortunately both a hemophiliac and a big fan of driving race cars, which turns out not to be a very good combination. He died after a car crash from internal bleeding. And then Queen Victoria's granddaughter, Alexandra, that's her right there. She married Nikolai II, Emperor of Russia, and they had their 5 kids. Olga, Tatiana, Maria is the one I can never remember. And their youngest, Anastasia, is right there. And then she's got her arm around Alexei, the crown prince, in his little sailor suit. And there's Alexei. He's the guy who would have become the next emperor, the next Tsar, if it hadn't been for the revolution of 1917. And he was hemophiliac because, just by stupid chance, he'd happened to inherit the X chromosome of Queen Victoria that happened to be carrying the hemophilia allele. And incidentally, Alexei survived, but every time he got a little cut, he was a major health crisis. And Alexandra was so desperate to help her son, he turned to a faith healer and a mystic who did seem to help Alexei with his prayers and meditations. It might have been the placebo effect, who knows? And he became very close to the royal family. His name was Grigory Yefimovich Rasputin. So if you've heard of Rasputin, if anybody remembers the old song, Ra Ra Rasputin, lover of the Russian queen, that was a cat who really was gone, that's him. That is Rasputin. So little minor way in which what we're learning interweaves with history. See, this class is a terrific value for money. You're getting multiple classes. It's not just biology, it's history too, at no extra charge. And it's 10:50, so that's a good place to stop. So I will now stop the recording.